1, 2, 3, 4. Okay, let's talk a little bit about how neurons are going to transmit signals to other cells, which is really going to be one of the fundamental functions of a neuron. If this is going to be some method by which we are passing information around an organism, they really need to get that information outside the neurons and into another cell. And these junctions between the neurons and whatever cell they're passing this on to, whether that's another neuron or if that's a muscle cell or something like that, these junctions are going to be called synapses. So let's take a quick look here. What you're seeing over here to your right-hand side of the screen is kind of a basic cartoon model of a synapse. And just to label the basic parts that we're going to be having here, this top part is going to be the transmitting cell. So this is the neuron that is going to have, yes, transmitting. This is going to be the neuron that has the information we're going to want to get into some other cell. And then down here, this is going to be the receiving cell. Now, again, this could be a neuron or something else. And then this space that is in between these two cells, this area of interface between them, this is the synapse. And that is what we're going to be talking about for the rest of this section right here, how that functions. So let's talk a little bit about the terminology that we're going to be using about various parts of this synapse. So the presynaptic neuron is this neuron that is sending the information. This is the one that is coming before the synapse. And so anytime we refer to anything being presynaptic, it's on this side. It's on the sending side of that synapse. So that is a presynaptic neuron. We also have the postsynaptic cell, which is going to be this right down here. Again, we don't call it the neuron necessarily. It could be a neuron. We could have a postsynaptic neuron, but they're not necessarily neurons. This could be a lot of other types of cells as well. We also have the synaptic cleft, which is also called the synaptic gap sometimes. And that's just this gap that's right in here between these cells, in which a lot of the action of this synapse is going to actually be taking place. The synaptic knob is this widening. At the end of the axon terminals, we have this widening area where all the kind of the functioning of the presynaptic neuron is taking place. And that's taking place right here. And that widening is known as the synaptic knob. We also have the subsynaptic membrane. In the postsynaptic cell, the membrane, the part of the membrane that is right below the synapse, is what's known as the subsynaptic membrane. So if we ever talk about that, subsynaptic membrane, it's that membrane in the postsynaptic cell that is right below the synapse. Also, we're going to talk a little bit here and there about synaptic delay, and really it's going to take some time to get whatever electrical signal that we have up here in the presynaptic neuron, to get it across this synapse and turn it into an electrical signal in the postsynaptic cell. That's going to take some sort of time. Or it doesn't necessarily need to be an electrical signal, it could be some sort of other signal as well. But whatever delay that is of getting this message across, that's the synaptic delay. And so finally, there are two major types of synapses that we're going to talk about, one a lot more than the other. And the two types are electrical synapses and chemical synapses. So let's go ahead and just talk really quickly about electrical synapses. And we're not going to talk much about them after that. Electrical synapses are kind of unique because right over here, they have gap junctions that are between the presynaptic neuron and I think these are essentially always between neurons and the postsynaptic neuron as well. So one thing that you'll probably remember from cell biology is that gap junctions have this unique feature that they're actually connecting the cytoplasms of the cells that's connecting. And so one great thing about electrical synapses is that they're going to allow ions, such as sodium, that would be up here in the presynaptic neuron, to diffuse right through here into the postsynaptic neuron. So if you have some sort of potential that is moving down the presynaptic neuron into these axon terminals, it can diffuse right through these gap junctions into the postsynaptic neuron. It's a really simple way of passing a signal along. Now, this has a couple really big advantages for cells that use these. First off, whatever potential that you have up here in the presynaptic neuron is going to be transmitted in essentially the full strength to the neuron that's receiving this. So that's one thing that's going to be different about electrical synapses and chemical synapses that we'll learn about a little bit later. The other really big thing here is they have very little synaptic delay. These are very, very fast methods of conduction. And so what you can have happen here is just many times faster move an electrical signal from one side, the presynaptic neuron, into the postsynaptic neuron. But there are a couple downsides of this as well. Really, there is only one type of signal that you can move from one neuron to the other. It's only going to be this potential that you can move across, this membrane potential. And really, you can only move the same kind of potential. You can't turn it into some other kind of potential on the other side. So that's a drawback of one of these. You have very limited ways that you can be passing information between these two cells. Not very complicated. The other thing is if we have some sort of signal that's coming along the other direction, that can move backwards as well through these electrical synapses. They don't transmit information only in one direction. You can be moving information in both directions. Now, more functionality, that seems like that could be a good thing. But actually, it turns out, in most neurons, in most synapses, that's actually a bad thing. You want to be moving information from one side to the other. You want to be moving in one direction. And so, the ability for electrical activity to be flowing backwards through the synapse is actually something we don't want to see and can cause a lot of problems for these synapses. Okay, so let's move on to chemical synapses. Didn't really talk too much about the electrical synapses because they're really simple, and there's not a whole heck of a lot to them. There's going to be a lot more to the chemical synapses, so this is going to take quite a bit longer. So, what chemical synapses do is they're going to cause this signal transfer by some chemical messenger that's going to be sent from the presynaptic neuron to the postsynaptic cell. And that chemical messenger that we're going to be sending across is going to be called a neurotransmitter. And commonly, these neurotransmitters that are sent from the presynaptic neuron to the postsynaptic cell are going to be amines, these small molecules that have an amine group on them. And you'll probably know some of these by, having heard of them before. For instance, serotonin, dopamine. These are kinds of things that actually come up a lot in pop culture. So, that's commonly what's happening here. And really, there are two important types of neuron-neuron chemical synapses that I want to be talking to you about specifically. The first of these are fast synapses. Fast synapses are defined because these neurotransmitters that are going to be released by the presynaptic cell are going to bind to ligand-gated ion channels. So if you remember back when we were first talking about membrane potentials and different ways that we can get ions through a membrane, one of the ways we're talking about is these ligand-gated ion channels where you have some sort of ligand, like a neurotransmitter, that is going to bind to these ion channels and cause them to open. And really the main kinds that we're going to see in these kinds of synapses in the subsynaptic membrane is going to be cation ion channels, which are just going to generally let any positive charge ion, small positive charge ion through. We'll also see some potassium ion channels and some chloride ion channels. Cations are going to tend to cause depolarization. And if you remember again, back when we were talking about graded potentials and the ways that we can change a potential, we can both have these as depolarizing, but we can also have them be hyperpolarizing. So some of these ligand-gated ion channels are also going to be potassium channels that will cause hyperpolarization in the cell, and then also chloride ion channels that will also cause hyperpolarization. We'll come back and we'll talk more about what kinds of things those do for this kind of transmission in just a little bit. The other kinds of synapses that we have are also these slow synapses. Slow synapses are ones in which these neurotransmitters that are emitted by the presynaptic cell are going to activate some sort of second messenger system. So they could be, now that second messenger system, as you'll remember probably from previous biologies you had, like cell biology, these can have a plethora of different actions. And so these slow synapses cause a lot of different changes in the postsynaptic cell. They could be just opening ion channels, but we want to open them for longer than these fast synapses can act. That might be one action of these slow synapses. They can also do some really crazy things, like actually change gene expression in the postsynaptic cell, or other causes like that. So generally, slow synapses, because you're using this second messenger system, are going to have slower actions, of course, but these actions are also going to be longer occurring as well. So we'll come back and talk just a little bit more about those later on as well. So let's sit here and talk a little bit about what's going on in a chemical, a chemical synapse. Let's just look at what we have here. Again, this is the presynaptic neuron up here. And then down here we have the postsynaptic cell. And then a couple different things. These white circles in here, these are going to be axonic vesicles. These vesicles are going to be holding neurotransmitters that can be released at any time. Also, surrounding this, and a lot of other places in the neuron, we're going to have these calcium ions out here. And this green channel right here, this is a calcium ion channel. Down here, these channels in blue down here are just, whatever they are, they're just some sort of ligand-gated channel. I'm not going to be really specific about what it is right now. And this orange thing in the middle, I'll talk a little bit more about that later on. Just accept that's there. Hopefully it doesn't bug you too much in the current time. So, this is what's going to happen during some sort of chemical synaptic transmission. The first thing is we're going to get some depolarization that is heading down, like an action potential, heading down this axonic terminal. When it reaches this synaptic knob down here, this is going to cause these voltage-gated calcium channels over here. I should mention that's what these are. These are actually voltage-gated. It's going to cause them to open. And because the inside of these neurons have this negative potential, we are negative, have a negative electrical environment just inside these neurons. And calcium has a 2+ charge, so it's actually more positively charged than either sodium or potassium. It's going to have an electrical gradient into the cell. Also, it's higher concentration in the extracellular fluid, so there's also a chemical gradient pushing inside. So these calcium ions are going to flood into the synaptic knob very quickly. And this flowing into here is going to cause these vesicles to bind to the membrane up here, the presynaptic membrane, and release this neurotransmitter into the synaptic cleft. So all this little purple neurotransmitter is going to be released into this synaptic cleft, and they're going to diffuse across it, and eventually what they're going to do is they're going to bind to these postsynaptic ligand-gated ion channels. And when they bind to those, it's going to open those up, and that's going to in some way change the membrane potential, because now we're letting all these ions through this membrane. It's going to change the ion potential, or the membrane potential of this postsynaptic cell in some way. Again, we'll come back to how that can happen in a little bit. Now, here's one thing that you need to pay special attention to here, is that this action on the postsynaptic cell is going to continue as long as there's neurotransmitter in this synaptic cleft. So what we need to do is to stop the action so we can get a repeated signal, is we need to get rid of this neurotransmitter as quickly as we can after it makes its action. And so there's a couple different ways that can happen. First off, neurotransmitter can simply diffuse out of the synaptic cleft. It can just diffuse away as one easy way of getting rid of it. Another thing that can happen down here is oftentimes there are these enzymes on the subsynaptic membrane that are specifically made to break down the neurotransmitter, and they're going to break it down. One thing to notice here is these ligands, these neurotransmitters, are only going to be bound to these ligand-gated channels for a very short time, less than a millisecond. They're going to open it up, and then they're going to pop off. And as soon as these neurotransmitters pop off, they could potentially bind with some enzyme that's going to break down that neurotransmitter. So even though we have these ligands binding to these ion channels, they're also going to be popping off and binding to these enzymes and breaking down. So we have these enzymes that are going to be breaking down neurotransmitter. The third and final way that we can get rid of this in here, actually have a reuptake transporter that is going to suck it back up into the presynaptic neuron. So this could be scavenging some of the some of the neurotransmitter that just used, and kind of recycling, using it over again and sucking it back up into there. So this whole action to turn some sort of signal, usually electrical signal, in the presynaptic neuron, convert it to a chemical signal, and then reconvert into some other sort of signal on the postsynaptic neuron, whether that's electrical or something else. This is actually a fairly slow action in terms of signal conduction. This is going to take about half to 1 millisecond to conduct that across this synaptic membrane, or this synapse. So the synaptic delay on these chemical synapses is quite a bit higher than what it was on those electrical synapses that we're talking about just a little bit